One of the biggest events in Europe in the biotech sector is without a doubt BioEurope, and this year's event was held in the beautiful German city of Leipzig. As well as lots of colour-coded booths for one-on-one meetings, there were also plenty of exhibitors, so Labiotech had an opportunity to find out about the biotech scenes in Portugal, Greece and Mauritius, as well as to talk to more than a dozen companies about their work.
Yeah, so Mendus is a company aimed at the treatment of tumor recurrence. So if you look factually in what is the major cause of cancer-related deaths, it's tumor recurrence. So we believe that that is one of the main topics that will also have to be addressed in order to make, let's say, the next move of innovation and the next major impact in cancer treatment possible. What we have discovered is that if you provide patients with a vaccine, and of course the design of the vaccine is a topic in itself, but to start with the basic principle, if you provide patients with a vaccine at the moment they have undergone first treatment, you have a good chance of boosting the immune system to control the residual disease. And that makes intuitive sense, but we also have thought about the actual therapeutic positioning. And also whether it is possible or not to design clinical trials around it. And with certain tumors, we are now with the lead program in AML, which is a very aggressive blood-borne disease. The recurrence is so fast that first of all it's a very high unmet medical need, but secondly also it provides for a clinical trial design that is aimed at recurrence-free or relapse-free survival. So what we have done is we have applied our vaccine to cancer patients that have undergone successful initial chemotherapy in AML. We look for patients that have a very high risk of recurrence because they have so-called measurable residual disease, or MRD, and that is a predictor of very fast recurrence. So those patients we treated with a very potent vaccine. It's actually a cancer cell-based vaccine. We administer it intradermally, so via a regular injection into the skin. And what we're now starting to see is that these patients respond to the vaccine. Later this year we will have a reporting of the data of that trial, so that will be a very important moment for the company, obviously. And what we also do is we use our knowledge, specific knowledge about how we design the vaccines, which uses dendritic cell biology, for other indications. So we're trying this concept of trying to prevent tumor recurrence also in another indication, in ovarian cancer, where also tumor recurrence is very imminent. So the majority of women have the tumor back after initial treatment within 2 years' time. So also in ovarian cancer we are trying to apply this principle in order to prevent or delay the recurrence of the tumor after initial successful treatment. And we are finally also looking into tumors that are very difficult to treat, established tumors, where we administer our products intratumorally. So where we inject them into the tumor with the hope to basically make the tumor more immunogenic, but that's a separate approach. But that combines, let's say, our technology bases into different directions. So we have quite a rich pipeline with near-term readouts in a number of trials, most imminently the AML trial. So the company is about these two main principles. On the one hand, understanding the biology that we apply to make very potent vaccines, and on the other hand the positioning which is addressing tumor recurrence and trying to prevent basically tumor recurrence instead of treating it.
And how does the vaccine work? Obviously without giving away any trade secrets.
No, it's well published data. So the cancer cell is not immunogenic, but it contains a very wide range, it's very rich in antigens that could potentially fuel the immune system to raise immunity against the actual cancer. But the cancer cells that we use as the basis for the vaccine need to become more immunogenic, so they need to be better recognized by the immune system. And we found a trick to manipulate these cells in such a way during our manufacturing process that they start to look like dendritic cells. And dendritic cells have a very strong immunogenic property. They are basically orchestrators of the immune system. They activate all kinds of other cells like T cells and NK cells and B cells, so they are really rich in molecules on their cell surface to get other immune cells into action. And that is something that we have combined in an off-the-shelf product. So the cancer cells are our cells. We have a cell line that is ours and that we use for manufacturing purposes, so we can grow them to large quantities, and then we shift them to this dendritic phenotype, and then all of a sudden they become highly immunogenic. Of course, we irradiate them so that they are safe to apply. We don't want to induce cancer, of course, in cancer patients, but it's a very safe product, and it's a combination of a cancer cell being very immunogenic because of the dendritic cell biology we apply to it. We inject it in the skin, so the eventual therapy is pretty straightforward. And what we have shown is that in the skin, doctors see a redness of the skin where the product is injected, which is a relatively good sign, right, because it shows that there's immune activation. In studies we also take biopsies, so we can show that there's a lot of immune infiltrates, so immune cells moving to the site of vaccination. But what we've also shown is that in the skin there's a lot of antigen presenting cells, and they are actually the ones that digest the vaccine, so they recognize, let's say, the cancer cells as a sort of brother dendritic cell, and they start interacting with it. But the eventual result is that they have phagocytosed, so they've eaten the vaccine, they migrate out of the skin, and then they trigger the immune system of the patient. So there's a lot of underlying science, but the factual therapy is actually pretty straightforward. And that's also making it attractive. Because if patients have undergone previous lines of therapy, and they're usually pretty harsh for the body, in blood-borne tumors it's aggressive chemotherapy, in ovarian cancer it's a combination of aggressive chemotherapy and surgery. So these patients have a relatively poor health due to the previous lines of therapy. So if you want to treat them with what's called a maintenance treatment, it has to be relatively safe. And of course leading to relatively durable responses, so that you really have an advantage, a clinical advantage for the patient. So our product adds up to all of that. And in the end, for the patient and for the doctor, it's a relatively easy to administer product with also really benign side effect profile. So that's what it adds up to.
And does it prevent the cancer from recurring, or it stops the cancer that is recurring?
Well, it is something we of course need to see improve in clinical practice. And I also cannot preempt the data that we will present later this year, because we are still collecting them for presentation at ASH, which is a major hematology conference held in the US. But what we have seen, and I can use that as a comparator, in an already completed phase 1 study, is that the responses we've seen in patients are very durable. So if you provide for a chemical drug, or even, let's say, one-time treatments like antibodies, they tend to have a shorter effect on the tumor. If you, on the other hand, boost the patient's own immune system, you have a sort of active immunotherapy or an active vaccination, you have the chance of a more durable immune response, driven also by memory formation of the patient's immune system. Now, whether that translates in a complete response, which we have seen with some patients, that they have gotten rid of their MRD, what I referred to earlier, so you can measure residual disease. We've shown in some patients the vaccination leads to a complete conversion of the MRD status, meaning they have gotten rid of their residual disease. That's of course a very clear sign of benefit, which needs to translate into survival benefit. There's also patients that have gone sick immediately. So they were unfortunately already on their way to an aggressive recurrence, and then vaccination doesn't stop it. And then there's a middle category of patients that still have MRD, but that seem to be relatively stable, so in other words also benefiting from the therapy. And that is the picture we currently have.
And is it it's just a one-time administration of the drug?
It's a good question. We have a vaccination scheme that's almost traditionally built up from an initial vaccination and then a booster. And what we do is we apply 4 vaccinations bi-weekly. Then there's a 2-month period after which we give 2 booster vaccinations. And that is then, in the end, the final vaccination schedule.
And so where do you go from there?
Well, I think we first put the bar high for ourselves, which is in the current setting we have addressed patients with, let's say, a very high risk profile of recurrence. So the MRD positive population in AML patients has a proven very poor relapse-free survival prognosis. So if untreated, it's less than 3 months actually before the disease is back after initial treatment. So first and foremost, we need to show that this has a chance in that kind of very demanding patient populations. If it works as a monotherapy, it's a very good starting point to look for a broadening in the hemato-oncology field. So the next step is, first we need to show that in this very demanding patient population we can show real benefit, but then it opens the door to a broadening towards additional patient groups. So the next step will be, and we will disclose more about that of course as we are planning our next clinical trials, and we have extensive contacts with KOL networks both in the US and Europe to get a lot of feedback on what would be a good positioning for a product like this. So the next stage of development will be that we'll set out a clinical trial strategy that will allow us to move forward and also to broaden our presence in the hemato-oncology field.